Inside a quiet laboratory at the University of Padua, the air was thick with anticipation. Researchers leaned in, their faces illuminated by the cold glow of a monitor. For decades, medical textbooks had taught us a simple truth, that secondary tumors were chaotic. But as the 3D reconstruction flickered into life, that old reality shattered. What they saw was not chaos. It was a precise, lattice-like structure, hauntingly beautiful. Dr. Stefano Piccolo and his team realized they were looking at a hidden architecture of disease. The team named this phenomenon the Metastatic Trabecular Morphogenesis program, or MTM. They collected samples from patients, primary tumors, brain lesions, and liver growths. Using deep transcriptomic analyses, they decoded the genetic messages hidden within. The data revealed a startling truth. When cancer cells migrate, they do not just grow blindly. They hijack ancient genetic blueprints, the same used during embryonic breast development. The cancer was not inventing a new way to survive. It was stealing a code. It was stealing a code meant for the creation of life itself. With the help of AI-driven imaging, the researchers mapped these structures in detail. They watched as the cancer cells underwent a radical transformation. They abandoned the dense, solid growth patterns of the primary tumor. Instead, they began to build complex, three-dimensional trabecular networks. This was a sophisticated construction project. It was not a reaction to the environment. It was an innate, programmed behavior. The team then looked back at the primary tumors, searching for a warning sign. They found that the MTM program was already active in the most aggressive cases. These tumors were already packed with the seeds of these branching structures. The blueprint for the spread is already there, Piccolo noted. High MTM activity became a clear signal of a high risk for future metastasis. It was a breakthrough that allowed them to see the enemy's next move. The complexity of the network was the key to its lethality. They had finally mapped the architecture of the spread. The search for the master switches led them to three specific factors: ETV1, ETV4, and ETV5. In a controlled petri dish experiment, they put their theory to the test. When they deleted the genes encoding these factors, the transformation was immediate. The cancer cells stopped branching. They lost their architectural complexity. Further investigation pointed to the FGF signaling pathway as the engine. They had found the fuel and the architects of the metastatic spread. Testing a receptor inhibitor called futibatinib, the team successfully halted the tumors. This discovery shifts the entire paradigm. Metastasis is not a random explosion. By targeting the ETV, MTM, FGF axis, we may finally stop the spread before it begins.